Okay, good Friday morning, everyone, and welcome to the Backyard Naturalist. My name is Tim. I'll be your host today. I should not call myself host based on today's topic. My name is Tim, and I will be your knowledge guide today, coming to you. I hope I'm not a host. I just jinxed myself. Coming to you from the home office of the Urban Ecology Center in Milwaukee, Wisconsin, where we just experienced a fairly dramatic temperature swing. I was wearing shorts yesterday, and I can confidently say that I'm not wearing shorts today, nor will I likely wear shorts for this foreseeable future. Long pants season, I think, is here for a while now. But you never know, 60-degree weather in Milwaukee is possible, even in December. So today's featured entity, I don't know what to call them, is a diverse group known as viruses, which I will say, as a research scientist, do objectively suck. They make us feel miserable, they can kill us, they can cause chronic issues. And, thankfully, usually they just make you feel terrible. And so maybe in some twisted, strange way, if we understand them a bit more, if we understand the processes happening in our body when we're under attack, that might just provide a tiny distraction from the miserableness of hosting a virus. Not likely, but we'll give it a try. So we'll look into the story of the virus in episode 11 of season 4 of the Backyard Naturalist, the Taming of the Flu. But before we go into this miserable but fascinating world, I'd like to say that you all here today joining us or listening in, you all do not make me feel miserable. You make me the opposite of miserable. You make me unmiserable, or dismiserable, or immiserable, anti-miserable. I guess that's my way of saying I hope you understand how much I appreciate you and your support of the series. You're the best. You're way better than the flu. And I want to give that extra thanks to members of the Urban Ecology Center and Backyard Naturalist subscribers for your added support in keeping this program going. Your support allows us to invite awesome speakers like Jonathan Call, Dr. Jonathan Call, a UWM professor of atmospheric sciences, who will be joining us next week here to talk about a wonderful part of our backyards, the clouds. Should be fantastic, and we're really looking forward to him hosting the program next week. We have finalized the details of the regional eco-travel trip to Indiana. The primary goal of this trip is to experience the splendor of the Indiana Dunes National Park. The itinerary set will be traveling to this truly unique and breathtaking place. We'll be in the thick of these glacial sands that suck your feet when you try to walk in them, that have been shaped over time to produce dunes, some which reach 250 feet high. The area is interspersed with wetlands, with prairies, old growth forests, and was really the primary site for some of the most groundbreaking work that gave birth to the study of ecology. And then we'll also be taking a trip down to Jasper-Pulaski Fish and Wildlife Area. Now we can start to monitor how many cranes are there. We're getting into the low digit 3,000-4,000 in a field at the same time. And it's very likely, based on past years and trends, that when we are there, we will be able to see more than 10,000 cranes in a single field. So leave all of the planning and the transportation to us. We'll take care of you. We're officially holding spots for folks as of this week. So if you're interested, contact me right away. And if you are a regular here on this program, you know that I've been maybe religiously following the flurry of human missions in space, particularly this intense interest in getting people back to the moon. So last week, I went through each of the missions, and a couple of updates. The really cool lunar payload mission of Hakuto R has been moved back from November 9th to November 22nd. So hopefully that'll get under in the sky soon. And then the big Artemis mission that we've been following has been delayed again, but this time for only 2 days. So hopefully, fingers crossed, it will have launched before we meet again next Friday. So that's the update on the big things happening in the skies. Then we make the transition to the teeny tiny things that can happen and that can invade our bodies, and have been invading our bodies since forever, since life has been here. Pretty much there have been viruses. So what is a virus? They're simple and they're complex. On one hand, they're, structurally can be very, very simple. Conceptually, they can be very difficult to understand, to classify. But again, at the core, they're simple. So we'll start with the straightforward things that we know about them, and then move into a little bit into the more complex. So what we're looking at here is something called a virus, or a virus particle. Those two are kind of interchangeable. A virus particle isn't a piece of a virus, it's a virus. It's a single virus, also known as a virion. And a virion has either 2 or 3 parts. And at the heart, the core, I guess viruses don't have hearts. At the core of all viruses is their genetic structure. That's pretty much what they are. They have nucleic acids, and they're either in the form of DNA or RNA. It's important to understand the difference between the two, because that makes a difference later when we're understanding how the viruses work. So DNA, or deoxyribonucleic acid, is composed of 2 chains of nucleic acids, the classic double helix that carries the genetic instructions for all aspects of life, of every kind of life, from growth and development to reproduction. These are the Legos of your life. So when you open a box of Legos, the DNA is that instruction booklet that helps you make the Millennium Falcon. You and everything alive is defined by your DNA. Or does the DNA define you? I don't know. In any case, this is the pretty important stuff. So when life takes that Lego box off the shelf, the DNA is the instructions that will determine if it will make a human being or a slime mold. It makes the difference between making a giant redwood or a microscopic protist. And if the DNA is the instructions, then the RNA is the coder. So my kids are really into coding right now. The DNA instructions create the code, which is the RNA. And then the RNA travels to the ribosomes, which are the factories. And they translate information that make the proteins that are, those are the building blocks. So, but as I think about it, I actually think I need to do a do-over here. So the Lego designers at the corporate office, those are the DNA. They create the instruction book. That instruction book is the RNA, and that travels to you when you buy it, and then you become the ribosomes, which make the Millennium Falcon. I think that's a better way to look at it. Anyway, okay, glad we figured, or at least I figured that out. So RNA is much easier to say. It's ribonucleic acid. It consists of a single chain of molecules that they're created in the DNA. Then the RNA brings that information to the ribosomes, and the ribosomes make the protein. So I hopefully clarified that, or totally confused you. I'm not sure which one, but so viruses have at their core this nucleic acid instruction in the form of RNA or DNA. And that's really essentially what makes a virus, but these nucleic acids aren't just swimming around by themselves. They're not old enough. The DNA or RNA in a virus is encased in a neat little package called a capsid. The capsid is made of a protein, so is made up of protein. So you have the DNA or RNA in the middle, surrounded by a protein coat, and that's what's called the capsid. So if these are the only 2 parts of a virus, and for many viruses, this is it. This is called a naked virus. There are some viruses, though, that have an extra layer called an envelope that surrounds that capsid. So this is a lipid layer that surrounds the protein layer that surrounds the genetic material. And even though it's mostly a lipid layer, there are some proteins kind of that can be interspersed, and that can be really important. So you either have a naked virus with just the genetics and the capsid, or you have an enveloped virus which has the genome, the capsid, and then this outer layer, lipid layer. So here side by side are these two kinds of viruses. They both have the genome, the nucleic acid, they both have the protein capsid, and then the enveloped virus has that extra layer. Viruses can also take on very different shapes and levels of complexity, and scientists further group them into three major groups. So you have what are called the helical viruses, which take the shape kind of a tube. So you can see here a naked and an enveloped helical virus. That's one of the major shapes. And then you have your polyhedral viruses, known as the soccer ball virus or the popcorn ball virus. Same thing, just different shape. And then you have the lunar lander module virus, technically known as, these are grouped as complex viruses. So this is probably the most famous one that I remember from my biology textbook. It's, this virus has the capsid, but it also has a tail with a body and legs. The one that really does look like it's out to get you, to colonize you. There's no hidden agenda when you see this one. It's like, I am going to land on you and inject you. I've learned not to trust anything that has protein spikes in it. And so really the sole purpose of any virus, if you can call it a purpose, is to make more of itself. That is all it does, and the only thing it does. There's no growth and development. There's no eating or playing or studying. There's just one thing, which is really at the heart of the question: is a virus considered alive if that's all it does? And I'll just tell you right now, there is no clear answer to this question. For many scientists, for something to be alive, it has to be able to sustain its life. And by most definitions, the smallest possible thing that can be alive in a sustained state is a cell. A cell can be an organism, right? You can have a unicellular organism, and it can exist as a standalone living entity that is able to eat and grow and reproduce. And so for many, a cell is the structure, the only structure that can do that. And then a virus doesn't have those mechanisms, but for others, because it reproduces, it has an element of being alive. But the only way a virus can reproduce is by invading a cell, which is why most people probably don't consider the virus alive. If a cell is the smallest thing that can be considered alive, viruses are way, way, way smaller than cells. At least most of them are. The vast majority of viruses, they're 1,000 times smaller than a bacterial cell. And a virus can invade a bacteria. So you start to think of the differences between viruses and bacteria. A virus can invade a bacteria, but a bacteria can't invade a virus. A virus can measure 1/1,000,000th of an inch long. And that's really hard to compare something to. Usually I can say, like, hey, this bird is about the size of a football. It's hard to compare something that's 1/1,000,000th of an inch long with anything that we can understand. But I did find this cool chart, the relative size of particles. And you may need to squint a little bit, but the Zika virus, which isn't the smallest virus, you can see on the bottom of this chart, and then the biggest thing here is the human hair. So we're talking tiny, 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 tiny. So it's 1,000 times smaller than a bacteria cell, which unfortunately means it's 10,000 times smaller than a respiratory droplet that you put in the air if you're sneezing or breathing. So one little droplet that comes out of your mouth or nose can have thousands of viruses in it. So a virus is probably just too small to do all these other things that other living things can do. So it can't eat, grow, and reproduce on their own. And they use their tiny, tiny stature to do what they do best, which is to invade, to slip into the machinery of your cells through the back door. It's ridiculously small. They can only be seen with powerful electron microscopes. In addition to being small, they're incredibly abundant. Being both tiny and abundant means that in one drop of seawater, there's on average 10 million viruses, one drop of seawater. So we'll get back to the question about whether or not viruses are alive. We'll probably visit that a bunch, but first let's look at that process. The one thing that viruses do best, this is their hedgehog. This is the one thing that they really know how to do. They know how to invade a host cell, and they know how to reproduce. And there are two major ways that this happens, and mainly based on whether it's a naked virus or an enveloped virus. Most naked viruses have a two-step process to invade a cell. First, they need to attach to the cell. And they use that with protein receptors. And then once they're attached to the cell, they inject the genome. So they can't just invade any cell. There are specific types of cells that they can invade, and that's important, good and bad, that's important. So if you have a case where the viral capsid is compatible, or the viral envelope is compatible, depending on what kind, to the cell, then it gives it that opportunity to invade. Once it attaches, it injects that material. It just kind of moves right in, and its guts are spilled. This is essentially what happens with the soccer ball, or the scary soccer ball, or the scary lunar lander form. But then the other way that this can happen is essentially that the virus itself, and usually it's this envelope that allows it to do that, because the capsid is protein, the envelope is mostly lipids. Your cell membranes are also lipids. And so if they're compatible, this type of virus doesn't just land and inject, it lands and it kind of incorporates its own membrane with your membrane, and it becomes part of that membrane, which really allows it then to enter. It kind of opens the door, becomes part of it, closes the door behind it, and now it's in your cell, and now it can again spill its guts, so to speak. So whether it's through injection or absorption, we now have the viral genome, the DNA or the RNA. It's inside your cell, and now it's ready to rock and roll. So the key thing is to remember that the virus doesn't have the mechanism to reproduce itself. So really what it does, and the way most people describe it, is that it hijacks your equipment in your cell. You have equipment that allows for your cells to do the things they need to do, but the virus doesn't have it. So if the viral genome is DNA, then it goes straight to the nucleus, and then the nucleus is like, hey, wait a second, who are you? Let me see your ID. And the viral DNA says, we're here from corporate, let us in, don't ask questions. They kind of intimidate them. Nucleus is like, all right, go ahead, you look official. And then the DNA goes in and starts, by being there, it starts changing the instructions to produce the coding to make more viruses. It changes the ambitions and goals of that cell from what it was doing before to making more viruses. So then the DNA produces RNA that is coded to make more viruses. That goes to the ribosomes, which are kind of like the assembly factories, who are working hard, making all the stuff we need. And then all of a sudden they're like, wait, what you wanted me to make a virus? But they're just following orders too, and then our own ribosomes kind of turn against us and start making the things that will likely make us very sick. So if that's if it's DNA. If it's an RNA-based genome, they don't have to bother with going to the nucleus first. They can just take their RNA straight to the factory and say, make viruses. And then the ribosomes are like, okay, I'm just following instructions. And then they make more viruses. So we have a cell that has been hijacked. It's producing more viruses. The viruses assemble, they get bigger and bigger, and then all of a sudden there's too many of them and they start to run out of room. And at this point, one of two things can happen. Usually, and there's no really absolute terms, if it was a naked virus to begin with, they can just burst their way out of the cell, and they essentially destroy that cell that worked so hard for them, they just kind of say, all right, we're done, see ya, kind of scorched earth. And then hopefully they get bad juju because they just did that. That doesn't seem very nice. But at least some viruses have some degrees of respect, I guess you could call it. And these are usually the enveloped viruses. So after they've taken over your machinery, they kind of slipped in through your lipid membrane, and they'll kind of bud off in a similar way to how they budded in. So they merge with the membrane, then bud off, kind of build a new envelope for themselves out of your membrane, and they don't destroy the host cell. So they're like the people that break into your home and eat your food, and then wash the dishes and sweep before leaving, and they lock the door behind them. It's a bit less harmful to us, and much less annoying, because it's really the thing that makes us feel crappy are those destroyed cells. When the cells are destroyed, that's what makes us miserable. So if the host cells that are used by the virus and then destroyed, if they're all in your nose, all those dead cells then become, your runny nose. Okay, it's just that's what everything that you kind of get a layer of dead cells that your body's now trying to get rid of. That becomes the snot. The snot tends to make their way into the back of your throat. And if it does that, then that can become a sore throat because it's attacking those cells in your throat now. And then they can also work their way through your bloodstream to attack your muscle cells. And so that's what causes our muscles to ache. So those are the viruses that cause us to be miserable. But there's way more to the story than that. At least these viruses are like, I'm going to attack you and it's going to make you miserable, but they're like in your face and honest, and you know what you're getting into. Other viruses can be much more sneaky, which often makes them more deadly. They're kind of the conflict averse, they don't want to cause trouble. They're just gonna, work their way into your host DNA. And they're just like, they're kind of smooth talking. They're like telling you what you want to hear. They're a little passive aggressive. These are the viruses you need to really look out for. So the retroviruses, they don't just send their goons to do the dirty work. They're inserting their DNA into your own cell's genome. They do it quickly and quietly before your cells know what hits them. And so now when your cells replicate in their normal, healthy way, they're also replicating with that viral DNA. The virus doesn't destroy them before they can, replicate on their own. So your cells are dividing and growing. They're carrying along this viral DNA, and that's spreading. For the moment, it's not doing any harm. But then all of a sudden, instead of, having this, one attack on you, you have this whole kind of army that's waiting inside of you to attack. And then there's a lot of different ways that can go. It could be just part of you forever. But there's usually then a trigger, some kind of environmental trigger that is a signal for all of those hidden DNA to start reproducing, to start getting those ribomes to make more. So that could be like UV light that triggers the viruses, and it could be stress. So if you get stressed out, you can, and actually not the stress itself, it's probably like the cortisol, the hormone that comes from stress. That's believed that we don't know what we still don't know, I don't think. Larry, you can let us know more, but like for cold sores, which are herpes caused by the herpes virus, that's kind of with you, right? And you may notice if you have it that it's often triggered by stress. And so the herpes virus gets triggered by stress, probably by cortisol. And so it's always kind of lying in wait, and then will come and go based on whatever the conditions are. In some, what makes the AIDS virus so difficult is that it lasts in that long inactive state, and it can be for years before you even know that you are a carrier, which means you can be a spreader. And then they have another whammy in that, viruses attack certain cells, and the viruses of the AIDS virus actually attack the cells that are there to your immune system, that are there to help fight the virus. So that makes them even more deadly. So viruses have a lot of tools in their toolkits. And collectively, as a viral community, there are a lot of different ways that they can get you. So this will be a good place to take a quick break and review. Watch one of my favorites, the Amoeba Sisters, for just maybe a better recap on viruses than my Lego example.
Chances are, in your life, you've had the common cold before. It can cause sneezing and coughing and can make you feel awful. The worst thing about the common cold is that you can take medications to help with the symptoms, but you really can't treat the actual cold with antibiotics like you would a strep throat infection or an infected tooth. Why? Well, let's talk about what causes it: a virus. The common cold is caused by a virus. While all living things are made of one or more cells, see the modern cell theory, viruses are not cells. Viruses aren't technically classified as living things since they lack many characteristics of life, but there is some debate in calling them non-living as well. Medications like antibiotics attack bacteria like E. coli, which are living cells. Antifungal medications can be used on fungi like athlete's foot, which are made of living cells. Viruses are not bacteria or fungi. You can't classify a virus as a prokaryote or a eukaryote either because it's not a cell. So, what is their structure like? First of all, viruses are much, much smaller than cells. You typically are going to need an electron microscope in order to see a virus. Viruses come in many shapes. Our favorite one to draw is the bacteriophage. It sounds very fancy, but it's a type of virus that tends to attack bacteria. Despite this being our favorite to draw, know that the shape of viruses vary greatly. One thing all viruses have in common is that they have some type of genetic material. This genetic material can be in the form of DNA or RNA. Viruses usually have some kind of protein coat, also known as a capsid. The capsid can protect the DNA or RNA genetic material. Viruses can also have other structures. Some viruses package special enzymes with them. Some viruses have an outer envelope. These additions can be useful to the virus in virus replication. One thing that makes virus replication or reproduction unique is that they can't replicate without a host. And it's their reproducing that tends to be a problem for its host, whether the host is you, in the case of the common cold, or an insect, plant, this bacterium. Viruses tend to be very specific for the host that they're going to infect, but anything that is alive can be a host because cells have the machinery that viruses need in order to replicate. We're going to talk about two types of viral replication cycles. The first one is the lytic cycle. In the lytic cycle, the virus, remember very selective, attaches to a host cell. It often binds to a receptor that this cell has, which gives it access to attach. The virus can inject either its DNA or RNA, depending on what kind of genetic material it has, into the cell. Some types of viruses are actually taken inside the cell themselves. Now, you would think that the cell would notice viral DNA or RNA or an entire virus that has been taken in, but in many cases it does not. It takes the genetic material from the virus and it starts following the instructions, which in this case is very bad because the instructions tell it to make copies of the virus. The cell uses its own resources to start building. It starts making so many copies of the virus that it can cause the cell membrane of the host to rupture, explode, lyse. So what happens is now that these new viral copies, they can get out of the cell and go and infect other cells. This is known as the lytic cycle. By the way, the lysing of the cell membrane is a very bad thing for the cell. The cell cannot survive without its cell membrane. The other cycle is called the lysogenic cycle, and I like to remember this is the longer word, so I like to think of this as the longer, sneaky cycle. Viruses that go through this cycle tend to do the same thing at the beginning. They inject their genetic material, but this time the genetic material stays hidden in the host genetic material. What happens is that when the host makes new cells, it replicates its own genetic material and the viral genetic material. Then those daughter cells make new cells, and they also replicate their own genetic material and the viral genetic material, and so on. This is the lysogenic cycle. That may not seem like a problem, but it really becomes one if the virus gets triggered to go into the lytic cycle from that point, because then all those cells that had the viral genetic material can start assembling viruses. And as far as what triggers it to go into the lytic cycle, it can vary. It could be a chemical trigger, for example, or a lack of food for the host. Going back to the common cold, a lot of times it has to run its course. Your immune system will typically take care of it, and medications can help with the symptoms so that they don't feel as bad. But unfortunately, some viruses are extremely complicated, like HIV, a virus that can lead to AIDS. Like most viruses, it is very specific. It binds to a CD4 glycoprotein that is found on the surface of specific cells, such as helper T cells. The problem about this particular target is that helper T cells play an important role in protecting the body in the immune system. Since HIV goes after these immune cells, it can make a person that is infected with HIV vulnerable to other infections. There are medications that have been developed to help keep HIV from replicating as quickly, and there continues to be research into looking into ways to keep the virus from attaching. Because if the virus can't attach, it can't insert its genetic material. A problem that continues to be a challenge is that many viruses, including HIV, can mutate. So a treatment developed against a certain virus type may not work on a mutated form. So, with our talk about viruses, you might wonder, is there anything positive about viruses? Well, some viruses can play a useful role in gene therapy. Definitely something to explore more. Also, remember how we mentioned that viruses can go after other organisms, like insects, for example. A virus that targets pest insects could be an alternative to a chemical pesticide. One such example virus that you may want to explore is the nuclear polyhedrosis virus. Still, it's important to consider that whenever talking about any kind of pesticide, chemical or viral, there could be consequences in the ecosystem. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
One thing about this too is that it's that reminder that, humans are attacked by viruses, but like anything alive can also be attacked, not just bacteria, but like the grass, the trees, the frogs, slime molds, all those things. Anything that's alive can be attacked by a virus. So let me just that again. All right. So here's a quick sum up of the viral process. You're visiting your brother at Thanksgiving. The whole family's around the table. Your adorable niece, who's contagious with a cold, but nobody knows it yet, sneezes. And you say, "Hey, buddy, I know you're only one, but sneeze into your elbow next time." But in order to say that, you first have to inhale. And when you do that, you inhale an airborne sneeze droplet, which we know can contain several thousand viruses. They attach to the sinus of your nose. The virus attacks, reproduces rapidly, produces an army of new viruses, produces a lot of dead cells, which causes this fluid to flow, giving you a runny nose. The phlegm drips down into your throat. The cells start attacking your throat. They give you a sore throat. The virus maybe spreads to your lungs, gives you that wet cough, phlegm in your lungs, and then into your bloodstream, where it can attack your muscle cells. And so when you're achy, that's what's happening. But thankfully for you, by this time, your immune system has also started to respond, hopefully. And now you're producing chemicals called pyrogens that cause your body temperature to increase, producing something we call a fever. And a fever can be very important because what it's doing is it's slowing down the viral reproduction rate. So most of your body's chemical reactions are optimal around 98.6, give or take. And then so as temperatures rise, these reactions that the virus depends on begin to slow down. And that gives your immune system a little bit of more time to kind of catch up and start eliminating those viruses from your body. So that's good. But then what's not good is that you're contagious during this time of fighting, even before you know it. One of the things I always hear from people is like, "Oh, it's okay. I'm no longer contagious." But a flu virus can remain contagious for a week after you realize the symptoms. And for both colds and flus, you can be contagious before you know it too. So then you sneeze before you realize it, and then the cold you got from your niece you spread to your coworker, and then so they become infected, and then the cycle perpetuates. Hopefully after you get your symptoms you stay home from work. And then so here's our second video, our last video today, from good old Josh here.
Hi, and welcome to Don't Be Dumb. I'm Josh Clark. Have you ever talked to a bunch of different people with colds and the flu, and they all say different things about whether they're contagious or not? Well, most of those people are dummies who don't know what they're talking about. You should listen to me, because I will tell you straight up what's true. See, when you become infected by a cold or a flu virus, it enters a few of your cells and uses your cell's own processes to replicate itself. This is called the incubation period. After enough copies of this virus are made, the cell is overwhelmed and it blows up. And the viruses are released throughout the body to infect other cells. This is when the contagion period starts. During this contagion period, all of the virus replicants are spreading throughout your body, infecting other cells. And those cells can be transferred from you to somebody else, like through your spit, or your sneezes, or your mucus. That's what makes you contagious. The incubation period lasts for a couple of days, and then when your cells erupt and you become contagious, you can be that way for up to a day before your symptoms show up. So you're contagious for a full day before you know you're sick, and then you stay contagious for several days after that. With the flu, you're contagious from the day before you show symptoms for about 5 to 7 more days into the sickness. With a cold, you're contagious the day before up to about 4 days into the sickness. So the next time somebody is sick and they give you some jibber jabber about not being contagious, you set them straight and tell them Josh sent you. Thanks for joining me.
And then again, we learned in the previous video that you can't fight a viral infection with antibiotics. So the tool that we do have to fight viruses, though, is immunizations and vaccines. Immunizations have changed over the years and how they work, but essentially what they do is they're kind of pre-infecting your body and getting your body to produce the right antibodies so that your immune system is primed and ready to go if the real virus hits your body. And then again, because viruses can reproduce so quickly and mutate, then we just have to update the vaccines on a regular basis. So there are a lot more topics to be covered about viruses. We'll probably revisit again in the future. But one thing that is not resolved and may never be resolved is whether a virus should be considered alive or not. Whether you think a virus is alive or not alive depends on your definition of what it is to be alive, and that's also not an easy one. And part of it is just semantics, right? What does it mean to be alive? What does it mean to be sentient? How many licks does it take to get the center of a Tootsie Roll Tootsie Pop? But when I'm knocked out by a cold or a flu, does it really matter whether the thing that's attacking me is alive or not? I don't care. What I'm thinking about is what Netflix show am I going to binge next to try to get me through this grueling period? That's when I need answer. And so I'm comfortable knowing that viruses might be alive, might not be alive. There really are organisms that are on the edge of life. And actually some of the stuff that I didn't get into today, that one of the videos alluded to briefly, is that there is a lot of beauty in viruses, and viral DNA is a major part of us. We're carrying around all this legacy DNA of our species and species before us. You have millions of year old DNA from viruses in your body. And then there's some processes like pregnancy that are likely only possible because of previous viral DNA. And because I said that, I think I am setting myself up for a part 2 in the not so distant future. So this was viruses 101, the basics. Viruses 102 or 201 will be the magical and strange. So they're incredibly simple. Again, just genetic material wrapped up in a teeny tiny package. But what they lack in size, they make up for in effect, because they've had a major impact on life for millions of years. They continue to impact us today, and they will for as long as life exists. So thanks for joining me today. I hope you can join us next week for Professor Call's talk on clouds, and we'll see you next time. And I will stop sharing my screen.